mean?
That's a good way to start. So we start off with Antonio's question for Sumaira about whether the phenylpropanoid pathway and biosynthesis pathway that you found could be related to selective pressures on bud development as they are in other gymnosperms.
So I'm not entirely sure whether they are related to bud development. That would but I'd assume that the best way of determining that would be to see to identify the genes in the other first, and seeing whether those genes bear any sort of sequence similarity to the genes identified as involved in phenylpropanoid biosynthesis pathways. But we do see that some of them are under selective sweep. So there's some evidence for that. I'm just not sure whether they're actually involved in the bud synthesis pathways.
Okay, and so kind of carrying on with the theme of your work for a second, from Tal Shalev at the University of British Columbia, why do you think the overall Busco completeness is fairly low despite the high continuity of the genome? And could there be errors in some of the context?
That's a really great question. And for a lot of this, actually, if we look at the transcriptome level, if you just assemble a transcriptome, if you saw in my talk, when I initially concatenated these 3 different transcriptome sources, clustered them, I had a Busco completeness of 98%, which was amazing. However, but when we aligned them back to the genome, because the genome is so fragmented, a lot of those transcripts seem to drop out. And that's where you end up with a Busco completeness of 68%. And we saw similar scores at the genome level too. And we think this is probably because a lot of these genes are going to have really, really long introns, which really can't be modeled in how Busco is predicting those orthologs. So that's one reason. The other reason also being that, as far as I know, the Busco database, the database that they're using, is not really representative of any gymnosperms. And that could also be maybe playing a role into why we have such low scores at some times. So that's my hypothesis.
Okay, yeah, that makes sense. Gymnosperms don't really fit into any conventional boxes. So next up is another question from the session chat for Lionel. So Tav Shalev again was asking about Torrey pine, and do we know why the migration of Torrey pine is so restricted? Do we know how self-compatible it is? And so this is actually relates to one of my questions, because you saw the population expansion apparently in the kind of semi-distant past. And I wondered whether that was related to the lower sea levels during glaciation and greater population extent.
That's actually a great question. There's actually, when it comes to demographic history of Torrey pine, I obviously run those models, but there are like a lot of different hypotheses out there for Torrey pine. Lower sea levels and a bridge between, I mean, I don't know how familiar you all are with like this region of California, but you know, you have the coast of California, and then you have the Channel Islands, and it's still really debated whether or not it existed or not. But some people have hypothesized that because of lower sea level, you might at some point have had a bridge between the super island Santa Rosa, which would be like all 3 all 3 Channel Islands, and the mainland. That would potentially have permitted the gene flow between the mainland and the island. So that was one hypothesis. Unfortunately, like as I said, it's really debated with whether this bridge actually existed. There are some evidence saying that it existed. Some others saying like no, it didn't. So it's not really really clear whether or not this could have been a factor. Other authors have actually said that maybe cone drafting in the water could have allowed like seeds to establish from the island to the mainland, as past currents were mostly from island to mainland. So you could have this gene flow occurring. Another, maybe probably more likely would be just pollen dispersal. Like pollen in pines can disperse like hundreds of kilometers. And although the island and the mainland populations are like 280-ish kilometers apart, it's not even that huge of a distance. So occasional long distance pollen flow could have accounted for that gene flow.
I suppose even if there wasn't a true land bridge, even if the gap was smaller, it
exactly. So here a bridge would not have been necessary. And we actually have also evidence that sometimes thousands of pollens can travel thousands of kilometers in pine. It's really rare, but it is possible. So with that 200 range, it's possible that pollen actually was exchanged between population. As it comes to self-compatibility, so far, Torrey pine seems to be it seems like selfing could be possible. However, you have a lot of mechanism, including the embryolethal system that would prevent self-fertilization in conifers, in pines and stuff like that. So I don't see, honestly, I don't have a clear answer to that question. I don't see why it couldn't be a selfer, but we don't seem to see a lot of selfing in Torrey pine. At the contrary, we like FIS estimates that I actually haven't talked about, I think, in the presentation, but we have FIS estimates that are actually negative, suggesting that you have more outbreeding that you would expect under Hardy-Weinberg equilibrium. So it seems like primarily Torrey pine seems to be like outcrossing, but whether or not it like selfing is possible, kind of hard to answer. I don't see why it couldn't, but
it's consistent with other conifers that we know.
Exactly, yeah.
Cool. Okay, question for Antonio. So I was looking at your paper and realizing you're working in a fruit tree basically, and although it's not necessarily a commercial one, I was pretty shocked by how close inbreeding really decreases fruit set. I wondered really what are the implications for that from like commercial fruit production?
You know, yeah, this is a very good question, and I have to say that first of all, the study species that I use in this paper is really close to Pyrrhus communis, which is the commercial one, and indeed in the Iberian Peninsula is pretty common to grafting this species, the commercial one, into the wild one because it's more resistant to drought.
Okay.
And also for Pyrrhus communis, it seems like it's important to have a good distribution of pollen donors because there are some potential problems about biparental inbreeding depression. So I think that these results are applicable to the commercial species.
Okay, and so to follow on from that, from Richard Snesko, how far apart would you recommend making tree selections for an improvement program in order to minimize relatedness?
That's a very good question. I mean, I think that we have to distinguish two different things here. One is if we want to have some recruitment within populations, thinking from the conservation viewpoint, no. In that case, more or less the genetic neighborhood, we are talking about 25 meters, between 25, 75 meters. So getting, pollen beyond these distances that can be covered by many pollinators is more than enough. One thing that we can do is, in terms of restoration, create genetically diverse neighborhoods. And this is something that we didn't in the past. So basically, we use a few genotypes and run the reforestations using these few genotypes, getting very genetically homogeneous neighborhoods. That is something that we should change. Then, in terms of how we can improve the reproductive output of the populations beyond the genetic neighborhoods, I think that based on previous studies where we didn't find strong evidence of local adaptation, we can bring pollen, or we can even bring seeds from other populations in terms of increase the genetic diversity and promote the vigor of the individuals.
Okay. That's cool. Interesting. I wonder what the implications are when you have a clonal fruit orchard.
That's a good point. And this species is also clonal.
But obviously, you can't have outcrossing there because you don't produce the fruit you want.
Actually, we have clones for Pyrrhus borgiana, and basically what you have is a functional self-fertilization. So you have trees in the field that you identify as different individuals, but if you only have clones in the neighborhood, basically you have no fruits.
Interesting. Okay, let's go back to Sumaira. What was it? Oh, I was going to ask a question quickly. Do you see any congruence between in your last plot with the network of interactions, do you see any congruence between the physical interaction and the co-expression of genes?
So from what I remember, there was obviously a lot more genes that are co-expressed versus actually having physical interactions. And a lot of those genes are actually not found in our annotation because, we're using Arabidopsis as a background. So far, I haven't been able to find an exact congruence, or I'm not exactly sure what I would expect if I were to look at the physical interaction genes versus the co-expressed genes. That's definitely an avenue to explore later.
Okay. Question for Lionel from Alana Mead. Have you been able to look at the potentially adaptive genes at all, and would it be possible to associate them with climate phenotypes? So this is Torrey pine.
Mm-hmm. Yeah, so that's really good question. So yeah, we've actually just started looking at these. So far, I mean, unfortunately, I've tried to annotate some of those genes by just blasting them against some protein database. Unfortunately, most of the information we have for pines, like Pinaceae families, is mostly a cytoplasmic genome, including mitochondria and chloroplast. So of course, I found a lot of these, but we also were able to find a few genes in the nucleus that, interestingly, were actually associated with defense against pests, including bacteria and fungus. And yeah, so far that's what we saw. We're still thinking about those results, but yeah. So for right now, we just have that kind of information. Personal observation is that we do have infestation by the bark beetle at the Torrey Pine State Reserve, so on the mainland. So something, just a hypothesis here, some speculation could potentially be that you do have actually selection on the mainland to try to fight off this beetles and try to resist to the impact of bark beetle. That's so far just a hypothesis.
Okay, cool. One further Torrey pine question from Natalie Isabel. Do they need fire to reproduce? What type of forest management would favor their persistence?
Okay, yeah, so not to my knowledge. They do not need fire, to my knowledge, to reproduce. Actually, the population on the mainland, a recent survey done by Travis with the Channel Island, actually suggest or show that you actually have an expansion of that population occurring on the island. So the island population seems to be doing quite well. When it comes to the mainland population, drought and beetle infestation seems to be the major causes of its decline. Like, you have a huge die-off of trees. You can see that right now. You have like patches of dying trees because of drought and beetles. So as a short term, I would say if you want to do a management that would favor the persistence, at least on the mainland, trying to regulate those bark beetles, which is actually underway with like traps, like pheromone traps, would be the best way of hedging your bets to actually improve persistence of Torrey pine in that population. Okay,
cool. Question for Sumaira. Do you think that genome sequencing projects should be focused on sequencing individuals, or should they be sequencing populations at low depth? This is from Eddie Lauer, sorry. He's thinking about whether you could use recombination to place sequence contegs into linear pseudomolecules.
So this greatly depends on genome size. As with Coast Redwood, the genome size is just way too big, I think, to be sequencing individuals. Although I think there has been recent attempts at doing this. Amanda Della Torre has been working on sort of doing an exome capture kind of technique for specific individuals of Coast Redwood. I think there's different questions we can answer when we have this kind of data, but at the initial attempt, we definitely need a good reference genome, an annotated reference genome, to be able to put the context of the information obtained from like exome capture data or population data.
Thanks. Let's go with the last question for Lionel. From Mung Mung Lu, how do you distinguish gene flow from ancient ancestral genes?
Oh, here, yeah. That's a very good question, and I actually have I didn't do it because yeah, I just couldn't do it with the information I have in hand. As we don't have a great genome for trypine, I had to assemble those loci using a de novo approach. So it was really, so there was no way I could know which was the ancestral state or the reference state. So in this case, we just work with minor allele frequencies. So that's a good question, and that would be actually something super interesting to know, like how much of this shared genetic diversity between population would be due to ancestral shared variation, or just like gene flow between populations. So that's a great question. Unfortunately, with what I have right now, I can't distinguish between the two.
Okay, thank you, everybody. I think with that, we're at our time limit, and we'll finish there. Thanks to Antonio, Lionel, and Sumaira.
Thank you very much, Ian. Thank you very much, guys.